Record.
All right.
Welcome to the diner. Thank you, Dr. Amber Van Laar and Dr. Victor Van Laar. They're very busy. They take time out of their day. Thank you. And they'll be talking about genes, gene therapy, and the differences with genetics. And wait, gene therapy and genetics. Yes.
Yeah, I mean, actually, this is why we're doing this. I think it's confusing, and we're gonna go do some like biology 101. So hopefully, some of you remember from back in high school, maybe. But go ahead and advance, honey.
Hold on a second.
Okay. So we need to make sure we're speaking the same language. So, I think, want to just make sure we're starting from the same page. And I think this was a request that came from, after I had done the talk for the symposium a couple weekends ago. And I think this is to get a better understanding of, what is gene therapy? I think you need to know what genes are to get to the point where you really can understand gene therapy. Please ask questions as we go through this. I wanted to talk about what are genes, what are genes related to Parkinson's, also what's gene editing, and then also dig into a little bit of gene therapy. But before we get into any of that, we need to just talk about what are genes. So just really simply put, genes are DNA. Your DNA is made up of lots of different genes. And the genes are the way that humans and all organisms on this planet can basically pass down that information from generation to generation. And so where is your DNA? So our bodies are made up of lots of different types of cells. Every cell in your body has a nucleus. So there's the center, the core of that cell. And that's where all the DNA is kept. And so the body has been really smart. It contains all that DNA really concentrated in the nucleus. And this is one way to protect the DNA. It's also one way to make sure that when we need to use the DNA, it's there and the cell knows where to go to access the DNA. Now, inside the nucleus, that's where the chromosomes are. So I think many of us may know about chromosomes, maybe at least heard the term before. And typically when you take these chromosomes out, they look like an X. And this is where we talk about females being XX. This is part of where that came from. Now, inside your cell, there are 23 pairs of these chromosomes. You get one from your dad, one from your mom. And depending on a whole bunch of different factors, it determines if it reads the mom one or the dad one. And this is really what determines a lot of, how we look and who we are in a really physical way, but also even deeper than that. And I'll go into that in a minute. But in each chromosome, I think that you can try to appreciate from these graphics here, you have to imagine there's just tons and tons of DNA wrapped around a core structure to make that chromosome. So there's a ton of DNA on this one chromosome. Typically, each chromosome contains DNA that has a different set of genes. And so there are some chromosomes that are responsible for different aspects. And this is one way that we can kind of separate out where the genes go. And this helps us understand, when mutations happen and cause disease, this is one way for us to understand and, where the problem is. So the DNA is, again, part of the chromosome. And the gene then is a piece of that DNA. So it's just a little section of the DNA. That is what a gene is. And it's the genes that then get read by the cell in order to then do something with that information. And just very briefly, I'm happy to dig into this a bit more, but DNA itself is made up of a bunch of other chemical structures that are then put together. And then because of the way that they are made chemically, they make a certain shape. And this is the double helix structure of DNA. That's really just because the electrochemical properties of these chemicals when they come together. All multicellular organisms, so this is humans and mammals and fish, and we all have the same basic structure of our DNA. And we use the same chemical bits that come together to make our DNA. So this is just a quick overview of what are genes. Any questions on that before I go on? A lot of this wasn't really known until like the 1950s. So just to kind of give some perspective here. Go ahead, next slide. So again, genes are, this is the inheritance. This is how we pass on just the history of who we are and how we've evolved as humans. This is how we pass that genetic information on from, parents to offspring. And this is really what makes us human. This is really how we have all the diversity that makes us human, but all the similarities that come with it too. And, this is, again, just remember, genes are DNA. DNA is made up from a bunch of genes. And again, you get one copy from your mom, one copy from your dad. And this is, again, where we get how you get blue eyes like your mom, or you get your dad's smile, you're as tall as your dad. I mean, things like this are really determined by your genes. And so I think traditionally, it's a lot easier from a genetic basis because they're to look at some physical traits. So again, things like eye color and even hair color. These are the things that are really just apparent just from looking at someone. But there are a lot of genetic influences on other things you cannot see. So IQ is an example. There is some genetic basis for that, but also other mental health things. There may be a risk for like depression, for example, that may be influenced by your genes. So things that you can't necessarily see. And then when it comes to Parkinson's, and we'll dig into this a bit more, it's even more complicated. And there's maybe multiple genes that are involved that make someone go on to have something like Parkinson's. Okay, next slide. So just some fun facts about genes and DNA. So, again, we didn't know what DNA was. We knew you could inherit certain traits from your mom and dad. It's pretty obvious when you have that baby and you're like, oh, that looks like mom, that looks like dad. But we didn't know that it was DNA that was actually doing that, and we didn't figure that out until about the 1950s. And once you kind of figured out that there was the structure and where it was in the cell, and that it was the genetic material that was being passed down, there was a big effort to try to understand that even more, and that's where the Human Genome Project came into being. And it was started in the 1990s. It took and didn't finish until just 2003. So we're almost 20 years actually to the day from when the Human Genome Project was finished. And this was their attempt to say, okay, now we know what DNA is, we know how to look at it, we know how to read it. So what does that look like for an entire human? And can we use that to then empower us to make new medicines and treat these diseases? And so this was a huge effort and really will revolutionize, and already has revolutionized, how we're looking at medicine and going forward from here. So another fun fact, if you took all the DNA from all of your cells and you just linked it strand from strand, end to end, 67 billion miles long, that would wrap around the moon 150,000 times. I thought that was really impressive. There was a lot of DNA packed into all of our little cells. Another fun fact, your DNA is over 99% identical to this person sitting next to you right now. So any two human beings on this planet have about over 99% identical DNA compared from one person to the next. And I found that to be just such an impressive number, because I think, with all that diversity that we all show and how different we all look and act and behave, it's really built off of less than 1% difference in our genome. I just thought that was really impressive. The other fun fact, when Jurassic Park was really, coming out, was a big deal. We are 97% similar to a T-Rex. On top of that, we have, tens of thousands of genes. So what makes us human is, I'm seeing averages between 20 to 25,000 genes that make us human. So just some fun stuff. Next slide. So I'm hoping some of you remember this from biology class, but when you have a piece of DNA, like, what does the cell do with it? So it's not enough to just have that information sitting there. The cell needs to do something with it. And so the cell needs to, what we keep saying, read the DNA. And it does this in a really standardized way in all your cells. So we start with DNA, and we have this little, it's a chemical, little protein that then runs along the DNA, and it reads it, and it spits out a copy of the DNA called RNA. And when you see that here, this mRNA, and then other chemicals come along and they read that copy, and then they make a strand of amino acids, and that is what a protein is. So again, the first thing is you run along that strand of DNA, you make this mRNA, and from that mRNA, you then have that read by other machinery in the cell. You make this chain of amino acids, and it's all those amino acids together is what makes a protein. And that protein is then what goes on to do all sorts of activities within the cell. So for example, in order to be able to receive the signal from dopamine in your brain cells, when it's the dopamine sitting out there in your brain cells, in order to be able to receive the message from the dopamine, you need proteins. That's the dopamine receptor, the transporter sees that. And that is all being based off of your DNA, and the genes that you have to make that protein. Signaling between cells. I mean, there's just so many proteins that are made every day in every cell of your body. This is happening in almost every cell in your body right now as we're sitting here talking. And this is the way in which your cell reads your DNA and to then do something with it. And the end result is really to make protein. So think of a protein as the business end of a gene. Any questions on that? Feel free to put things in the chat too as we go along. Okay, next slide. So, this is when things are working well, that this is how the normal process works. This is how genes are being read every day. But what happens when genes go bad and something goes wrong? The next slide. So how do genes then cause disease? And this is going to be relevant for us in our later conversation, and how is this linked with Parkinson's and how? So now you can see all the different components that put genes together, the different chemicals, the A, G, T, C. You can have an error where one of those letters is put in incorrectly, and that can then cause an entire disease that's in full systemic disease. Sometimes you can have an entire section of DNA that is missing or it's shifted, and that also can cause some types of disease. So you can delete things, you can shift pieces of DNA around, you can have extra copies of things, or things are just absolutely missing. I think a common example of this is like the Down syndrome, where you actually get 3 copies of one of the chromosomes, and that's what then leads to the development of Down syndrome. So there's lots of different ways in which things can go wrong. It's actually very impressive that all of us are sitting here right now, knowing how easy it is just to make one little mistake in reading any of those letters and putting the wrong thing in the wrong place. It's amazing that we are, as functional as we are as humans. Next slide. And so, just wanted to give some examples of what are some genetic diseases that you may have heard of before. So again, this is where typically a mutation happens. So there's something gets read incorrectly, and that protein is made wrong. And this could be a copy from mom or dad that was made incorrectly there, and that's what you inherited from your parents. But sometimes this can happen spontaneously, where there's just, for whatever reason, something was put together, in an incorrect way, and that then leads to mutation that goes on to causing problems. Now, typically, if there is a change that's happening in the DNA, that is the DNA that could be passed on to children, and that's why I think this is an important topic and things for us to think about. Because I think there's a lot of concerns with family members. And so this is where things like cystic fibrosis come in. So I think a lot of us have probably heard of cystic fibrosis. Anymore, they're actually checking for this. If you are newly pregnant and it's the first time you've been pregnant, they're actually offering to check for this because it's so common, and it's good to know ahead of time there's ways to potentially treat this. There's muscular dystrophy. So these are problems with muscles getting weak, and mostly affecting boys. But there's a lot of other genetic diseases you can see listed here, and hopefully you've heard of some of them. And these are typically based off of like one gene that's not working right, or there's a problem there. Now, there are a lot of other what we call multifactorial disorders. So they're things like Parkinson's fall into this category, where it's like, yes, there is some genetic links to the causes of Alzheimer's and Parkinson's and other neurodegenerative diseases, so other dementias, other movement disorders. But it's maybe not just the gene that does it. It may be some changes in genes plus the environment, plus maybe something else that then causes someone to go on to have something like Parkinson's. So heart disease, like having the coronary artery disease, this is another one where it's probably a combination of factors. You may be at risk because dad had a heart attack, but you're maybe not helping the situation by smoking, for example. So again, there's a lot of diseases that are considered multifactorial. Go on, next slide. So let me just talk now a little bit about genes and how this is influenced in Parkinson's. Go to the next slide. So early on in my training, very early, there was an idea that maybe there weren't any genetics involved in Parkinson's. Actually, for a long time, it was thought that this was environmental. You had to be exposed to something outside of yourself in order to explain why someone was getting Parkinson's. And it's been, a few years now, but it was really, I think it was a Italian family, if I'm remembering correctly, that there was just a lot of Parkinson's in this one family. And sure enough, when they went to go look, there was a problem with the alpha-synuclein gene. And so it was one of the first links where it was, I think scientists and researchers finally had that aha moment of, oh, gee, there's clearly some basis, some genetic causes here that may be contributing to why someone would go on to have Parkinson's. And that was really revolutionary. I think when we think about genetic diseases, this really changes maybe how we approach things from a treatment standpoint. And so over the years, now that we're starting to realize, that we maybe were naive to think that there was no genetic cause, there's now new genes being added all the time to the list that's being linked to Parkinson's. And so the quotes that we give now, it's estimated about 10 to 15% of Parkinson's has a really clear link to the genetics associated with Parkinson's. So that's actually a small sliver of patients have like a clear, it's clearly just genetics driving the diagnosis of Parkinson's. But in fact, there's actually a lot of mutations that maybe they don't suggest like 100% chance you're going to get Parkinson's. So there's one called GBA, and you can have a brother and a sister that both have the same mutation, and one gets Parkinson's and the other does not. And so I think that there's still a lot we need to learn about how Parkinson's influences, or how genes influence Parkinson's diagnosis. That being said, it's clear that there are some populations, there are some like in the Ashkenazi Jews, and there are some Berbers in Africa that may be at higher risk of developing. So clearly genetics have a role in Parkinson's. And I think we're just starting to scratch the surface on how it may affect for the majority of patients with Parkinson's. And so the way that we describe this now is that most people that have Parkinson's, it's actually a combination of both the genetics that they bring into the situation from their parents, but it's also the environment in which you live. And this includes, yes, exposures to toxins and pesticides, but it's also like smoking and caffeine use and exercise. And then it's just other things that in your environment that you maybe have no control over. And so the idea is that there's a genetic risk factor plus the environment that may cause Parkinson's to happen for the majority of patients that have Parkinson's. Next slide. So again, we're really still in early days. And I think one big change that's going to happen is it's a lot easier for us to test DNA now. Like we're way better at it. It's cheaper, it's faster, it's more commercially available. And with that, we'll have a much better understanding as to, how genes are involved in Parkinson's. And I'm guessing like that list keeps growing. Every time I look at it, the list of genes associated with Parkinson's just keeps growing and growing. And so I'm sure that there are other mutations we just haven't found yet because we just haven't had the chance to look far and wide enough amongst patients with PD. And again, just keeping in mind, and this is going to be a really important point, that even if you had a mutation, it doesn't guarantee that you're going to go on to have Parkinson's. That's not how I think a lot of these mutations will end up working. But it does give us some hint as to like what causes Parkinson's. And I think that that's really important for us to just better understand, how Parkinson's happens in the first place. And I just put a little shout out here. There's a big push right now to understand how genes are influencing PD. So the Parkinson's Foundation, the national group, they have this PD generation. And so it's this big, interesting study that they're trying to put together where they're taking DNA samples from patients with Parkinson's and their family members, and in a really big way, trying to collect this information to see if we can have a better understanding of, what causes Parkinson's, what may cause it to continue to progress over time. Other groups like 23andMe, and there's another company we work with, it's called Invitae. They all have panels of looking for risk factors for Parkinson's. And there are a lot of other companies that are starting up that are offering, cheap, fast, easy tests to look for risk factors for Parkinson's and other similar types of diseases. Next slide. I'm sure Casey can send this out to you guys later. I just wanted to give you a couple of websites to go look at that I thought were pretty decent. So the PD generation that I just talked about, this is with the Parkinson's Foundation. They have an entire really nice website actually about different causes and genetic links to PD. You're welcome to go check out. The Fox Foundation has also taken this on, as a big task for them as well. They have an entire section just dedicated to understanding the genetics of PD. The American Society of Cell and Gene Therapy, they have really great, just basic understanding of what are gene therapy basics and what are genes. And then there's actually a really nice section from Medline Plus looking at genes and Parkinson's. And I'm sure Casey can send these links out to you later. Any questions before I go on to the next section? Okay. So this topic comes up a lot. And, and I tend to just move past it because it really requires its own dedicated discussion. So this is becoming a little more popular, let's say, on the news, and this has been in the media a bit more. This idea of gene editing, this also became like a really big and a hotter topic. Back in 2020, their two scientists received the Nobel Prize for this gene editing technique called CRISPR. So I just want to take just a few minutes to talk about CRISPR because I'm guessing some of you at least have heard of this as a thing. Next slide. So let me just try to quickly go into what CRISPR is. CRISPR is one way to do gene editing. There have been lots of ways that we've been able to manipulate and change DNA. The thing about CRISPR that's been so revolutionary is it's allowed us to do it very precisely. It's been really hard to say, I want to change that one little tiny piece of DNA in a very consistent, repeatable way. We're able to now do that with CRISPR. You're welcome, Bill. And so this is what we are calling genetic scissors. So this is one way to go in, snip out the one little piece that you don't want or that you want to change, and then allows you to put in exactly what you do want. This is really, really powerful technology. And we've only started to scratch the surface on this. I mean, this is very early stages for this new technology. But one way in which this is being most frequently used right now is to develop models for different diseases. So in the lab, we were using this pretty frequently. We wanted to study and make like cells that looked like they came from somebody with a certain type of mutation. We could make that happen in the cells. So to really better understand, what the causes of the Parkinson's type would show up, and then look at different treatments and see if there's something that may work better for that particular mutation or not. And so there's lots of ways in which CRISPR can be used in this way. So again, you can use it to make a model of a certain type of gene mutation, or you could even potentially take some mutated cell type and then you change that mutation back to what is more normal, or what we call wild type, and see if you can correct the disease. That's one other way to use it. And so taking this really powerful technology It's one way to be applied in medicine. I think those are very obvious, but we can extend this to crops and other like livestock. I mean, there's just the possibilities are endless, and there are some potential consequences for this, which I'll talk about in a second. Next slide. And so, again, there's some controversy here. And so I'm going to just like touch on it today. I'm happy to dig in on this a bit more if anyone's interested. But, you know, one of the first ways it was being used was really in plants and plant science. And so, you know, could we make, you know, disease resistant crops or drought resistant crops? There was one of the first ones was looking at non-browning white button mushrooms. So they were able to genetically modify white button mushrooms so that they didn't turn brown. And that was one of the first types of plants that was being modified and was accepted by the FDA and all of this that you could get today. But I think, you know, for my interest, and I think for a lot of what we're talking about today, you know, how is CRISPR being used in genetic diseases or just as in a medical way? And so there's been a lot of work at blood disorders using CRISPR. So this is like sickle cell disease, certain types of hemophilia. A lot of this has been looked at in clinical studies already using CRISPR. That cystic fibrosis I just mentioned, this is a real problem for a lot of kids that are born with this. And there's really no good like medication you take by mouth to help with this. So you know, using a technique like this to correct this one little mutation may be revolutionary for this disease. Something like Huntington's, where it's, again, one gene is messed up. And maybe this is another way for us to approach this really disabling disease. But I think it's really cancer that's been taken advantage of CRISPR the most. And you'll see a mention of this a little bit later, but what they have done is they've taken immune cells out of patients that have a certain kind of cancer. So I think there's been a lot of, like leukemia being used, blood cancers, prostate cancer. There's melanoma was another one. So you take the immune cells out of a person's body, you do CRISPR to edit those genes so that they now know to attack that cancer cell type. You put those cells back into the person and then use your own cells to then attack the cancer. So this is a process that typically happens with cancer is your immune system should recognize the cell as rogue, that it's bad, it needs to be taken out. And so you're basically taking that known mechanism of attacking cancer, and now you're just like augmenting that. You're really making lots of cells that now know exactly, what they need to target and put those back in the body and let them do the work. So this means, you're not using chemotherapy. You're not, all those bad things that happen with chemotherapy are not a problem now because it's not chemicals. You're using its cells. And so there's some really interesting studies that are being done. And I think we'll be seeing more and more of this coming forward, especially for cancer using CRISPR or genetically modified cells, something else called CAR-T. I'll mention that here in a little bit. One other way CRISPR has been used with some ethically questionable use of CRISPR. There was a Chinese scientist who manipulated human embryos to make them resistant to HIV infection. This caused a worldwide uproar about the use of technology in this way. And there is still a worldwide moratorium on you cannot be using CRISPR to genetically modify like human DNA like this. So just keep that in mind.
Human DNA.
Thank you. Yep. Go to the next slide. And so, with great power comes great responsibility. I couldn't apply to any better technology than this. This is really, really cool stuff. And yes, we can make some really very specialized precision medicine this way. Biofuel, which is great. And these, crops that are now going to be, more, drought resistant or disease resistant. There's some really great things that we could do with this technology that are already being explored or already being put out into the world. But, let's keep in mind the challenges and the concerns that we have with this new technology. And I think any new technology that we're talking about, but the safety issues, we still don't know. I mean, this was 2012. I mean, this is all very, very new technology. And what happens if we genetically modify these cells? Sure, maybe they're okay in that first year. What happens in 10 years? These are things we just don't know yet. And so this is why we need more time to understand this a bit better. The ethical concerns, I've only just mentioned a few things, but there is for good reason, some serious concerns with, manipulating DNA. This is, where the designer baby concerns come into play, and, what makes us human, and some really, heady types of topics really start to come up. And it's those ethical concerns that are really driving the regulation of this technology. And so in every country has a slightly different perspective on this. And so it's going to be interesting to see how the story evolves over time. But just to kind of balance these things out, that we have some really cool technology here, but we need some more time with this. We need some better understanding. Any questions before I leave CRISPR? This is just a really quick snapshot.
There is a question, is CRISPR being used in leukemia?
Yes, lots of blood diseases. So leukemias, lymphomas. It makes a lot of sense to start there with CRISPR. So again, these are blood cells that are circulating all over the body. You're then putting immune cells back into the bloodstream. So the contact, the chance for them to interact with each other is really great. And so I think for a lot of these, we call liquid tumors. So they're like blood tumors, like leukemia and lymphoma. They're perfectly positioned for this kind of therapy, and it's being explored, and it's going like gangbusters right now.
So, yeah, but I mean, that's also another thing to emphasize is we're talking about things that are still being researched. There's no CRISPR in the clinic for anything yet. And the CRISPR tech itself is still evolving.
Let's go to the next slide.
All right.
So there is a collision actually of gene editing and gene therapy, but let's dig into a little bit of what is gene therapy, because I think that this is a definition that's actually quite broad. But one way I like to talk about it is that we're using genes as medicine here. So and you can do this in a variety of ways, but think of it as using genetics as medicine. Next slide. Some of you may have seen this before. You're gonna have to advance through this, honey. So again, going back to the very first slide, genes are DNA. It works as like our blueprint or instruction manual for our entire body. And we need this for the health and growth of all of our cells. Next slide. And as I showed you earlier, it's every cell in your body has machinery in place to read your DNA or read the instructions to figure out what to do with that blueprint. And it's really those proteins that are being made from your DNA that really is required for just keeping your cell alive and functioning and doing its proper job. Next one. And again, when we do gene therapy, we're taking advantage of that machinery that's there. The cell knows how to read DNA. This is what it does. And with gene therapy, we're taking advantage of the natural function and using genes that we want to then make the protein that we want. And that then acts as our medication. Next one. Next one. So keep going, honey, just click through. So again, genes, think of them as instructions for every cell in your body. And we can use this in a variety of ways. So we can use gene therapy to correct something that was like a mutation that's not working well. So you can now take something that's not working and restore its function. We can inhibit. So if you had a gene that's making a bad protein that's causing disease, you could turn it off. It's where you can inactivate the function. Or you could find something that's working well. Maybe you don't have enough of it, and you can enhance or modify the function of that certain type of gene. Next one. Just click through this. Next slide. And with gene therapy, and especially how it's being done for neurologic diseases, you take a copy of the DNA. So this is think of it being made in the test tube essentially. So this isn't coming from somebody. You know, again, we've had the Human Genome Project now. We know what the sequence or we know how to read the DNA for most of the genes that we're interested in. So we make that little piece of DNA, which is your gene of interest, and use that to treat the disease. So, but how do you get that into your cells? And this is where you need an envelope or packaging material. A lot of gene therapy as it stands today, maybe not forever, but right now we're using a viral vector. So you take this modified vector, put your DNA in there, and that's how we get our packaging so that we can get it into the cell. The packaging I'm going to be talking about more today is called AAV. So this is adeno-associated virus. So this is used by the company I work with, but a lot of other groups are using this one specifically also for brain cells, because it's really good at getting DNA into brain cells. Next slide. And you know, for me, this is the challenge I face every day when I get up. You know, this is a very short list of what's currently being approved and currently being used right now in the clinic of gene therapies. This is it. This is all that's available in the US. So there's Luxturna. So this is used to treat an inherited form of blindness. This was approved just in 2017. It's a very specific form. This tends to happen for adults. It has been really good at improving vision. It's not going back to normal, but it allows them to function more normally in their environment. The other one that's a little bit more recent is called Zolgensma. So this is used to treat, they're typically children who are inherit this SMA. And this is, you take kids who can't, you know, climb up a couple stairs that now are able to do a lot more normal things. And again, this was just approved in 2019. We have a lot more to do to be able to have more therapies out there and available for patients, other gene therapies for a more variety of patients. But there's one cool thing that's happening here, and this is the CAR T that I had mentioned earlier. This is typically being used for cancers right now. But again, you take these cells, you edit them in a way that you think is going to help the disease, and you reintroduce those cells back into the patient to then treat. So there's this combo of like cell and gene therapy that's also being explored. So this is just the advent of all this happening. Go to the next slide. Next one. Just click through. Now, typically, most of gene therapy has been used, and a lot of early development, we knew gene therapy should only be used for genetic diseases. And I think that definition has really been expanded a lot in the past few years. And so no longer do we think of gene therapy only for like known one gene's messed up type of diseases. And, you know, especially when we think about like Parkinson's, for example, there's some huge advantages to thinking about gene therapy for Parkinson's. And primarily because it's a one and done. The idea is that you can apply gene therapy directly to the brain and you don't need to do anything more. Like the brain cells are not dividing. You can't make new brain cells, which is part of the problem why we have Parkinson's in the first place. But that also means that this is a therapy that will be long lasting for someone with Parkinson's. And in the past, this has been done in a variety of ways. So you can use gene therapy to really boost the dopamine in the brain. So this is one strategy that's been used a lot, actually, to look at gene therapy in Parkinson's. But I think the holy grail here is can we use gene therapy to modify disease progression? Can we slow down the progression of Parkinson's? This is part of what I'm looking at in my current studies. Go ahead. So let me just talk a little bit more about AAV. Apologize if you guys have seen this before, but I think it kind of helps to see it again. So again, the envelope that we're using is this AAV. That's the kind of weird starfish looking red and purple blue doodad over there. So think of that vector as your packaging material. It's your envelope. Next one. And so just click through, honey. So the AAV2 is typically used for brain diseases. It's been used a lot for Parkinson's specifically. And again, the brain cells themselves really don't like taking up stuff. There is a lot to protect those brain cells from picking up, infections and things like this that you don't want in the brain. And so when you do want to try to introduce something new into a brain cell, it's really hard. And so unlike other cells where you can just have DNA floating around and it will soak it up, that doesn't happen in brain cells. And so we need some way to deliver it. And this is one method, is AAV. So it turns out that AAV is actually pretty good at getting into brain cells. And it's been used a lot actually in a lot of other brain diseases and brain cancers. So it's been safely done. And so I think we have some history and we understand it a bit better. Next slide. You're gonna have to, this one's animated. Go ahead. So kind of what we had shown before, think of your letter is like your DNA. This is your instruction manual. So the purple DNA is like the purple letter. In order to get it delivered, you have to put it in the envelope. So you package your letter into the envelope and now you need to address it and get it sent. Click through. And in the case of what we're doing and a lot of other gene therapies for Parkinson's, we use surgery that's like our mail truck to then deliver it to the right address. And it's then the brain cells that pick up that envelope, open it up and read the letter. And it's when they read that letter, they then make the protein of interest. And for us, this is something called GDNF. So we say, cell, I want you to make this protein because I think that's going to help this person with Parkinson's, for example. Next slide. Just another way of putting it, this is our DNA. We have to get it packaged. We have to put it in the envelope. And it's interesting, this actually can happen pretty naturally. They kind of like to come together. And then in our case, we actually put the gene therapy right into the parts of the brain that are impacted by Parkinson's. And we know that this packaging material is good at getting into cells. And what it does is it delivers the little gene. The packaging itself gets crumpled up, thrown away, and all you're left with then is the little piece of DNA or the gene that you're interested in. And again, what we're studying is GDNF. And so the cell knows what to do from here because it does this every day. It says, oh, there's a piece of DNA, I need to read that. And it reads it and it makes the protein. And in our case here, it's this green stuff is the GDNF. Next slide. So, I think that there's been actually quite a few studies going back to, I think, 2003. There have been studies looking at gene therapy for Parkinson's disease. So I think for me, that's a really impressive, historical perspective here as to how long we've been trying to do this. And really the two main methods that have been looked at to use gene therapy in Parkinson's is one, you boost the dopamine. And this is great because the idea behind this is that it helps you manage motor symptoms a bit better. And potentially lower medications and prevents the ups and downs that come, with later stages of disease. And this has been looked at quite a bit over the years. And think of this kind of like the same way in which DBS is working. It's trying to smooth out the ups and downs, maybe lower your meds, make things a little bit more easy to manage. So that's one method. Another approach is looking at ways to slow down disease progression. Now, this is really the holy grail for any neurodegenerative disease. So Parkinson's is true to my heart, right? But the hope is that you can actually capture someone that's in the early stages of disease and what we call the honeymoon period, when it's there, it's annoying, but it's manageable. This would be the goal. And the idea is that you would then prevent the onset of a lot of non-motor symptoms that are scary or just troublesome. So cognition, like the memory impairment, these sorts of things are really scary for patients. And, the ideal here is that you can stop the disease from moving on so that doesn't happen in the first place. That would be ideal. And I think the other hope is that it would prevent the worsening of the motor symptoms. So things just get more complicated in managing the motor symptoms, tremor, stiffness, slowness over time. And so if we could keep things like they were in the early days when things were easier to do, this would be an ideal place to use something that would be disease modifying. And again, we want to just hold symptoms where they are. If we can't get you back all the way to normal, can we just keep you where things are manageable? And now it becomes more like a chronic disease like diabetes. It's there, but you can live with it. We can manage it. This is really the holy grail for a lot of companies. Next slide. And I think it's the last one. Yeah. So, okay. That was a whirlwind. So I think there's some questions in the chat.
Yes, there are. So the latest one to come through. Do we understand what is killing the dopamine neurons in the first place? Excess dopamine inside the neuron. So I can take that one if you want.
Go for it.
But the short answer is no. We actually don't know what triggers the start of Parkinson's disease that leads down to the death of these neurons. By the time we're studying the loss of the neurons, we're literally looking at ghosts. We're looking at what's left behind to tell us what happened. So we have some clues that oxidative stress was increased. Dopamine was mishandled. So yes, the excess dopamine inside the neuron could be a contributor. We have alpha-synuclein accumulations, but we don't know if any of these, it's a chicken and egg question. We don't know if any of these caused the neuron to die or they were a symptom of the neuron dying from something else that we haven't figured out yet. So we don't know what's causing, I have to take a call. I'm sorry.
Go for it. I see. Yep, you go ahead. And I think that there's, maybe it isn't one thing, right? I think that there may be multiple things that need to kind of come together to make a cell die. But part of the problem is that by the time someone comes into clinic to see one of us in the first place, they've already lost like maybe half of their cells. And so we're already starting way behind the 8 ball. And I think this is one big reason why it's been really hard for us to get ahead of this problem. Let's try another question. Donna brought up this question about brain fables. It's a great book. I happen to know the authors fairly well. I think it's a really good way to understand why Parkinson's has been so hard to get on top of. There's a lot of history that they go through in that book that I think it's an advantage to anybody who wants to read it. So if you check it out, I would suggest. And then there's a question from Norma about, could you use gene therapy to treat things like lupus? And yes. So I think that there's a huge interest right now in looking in a lot of what we call autoimmune disorders as a way to use gene therapy. So I think you'll start seeing this a lot in the news coming up. But yes, there's a lot of potential there for that. And then Bill said, since the gut and brain are linked, wouldn't it be nice if you could introduce the gene therapy in a milkshake? So I didn't go into this right now, but there's a lot of interest in, I would like to get away from having to put gene therapy right into the brain. There is a lot of effort being made right now so that you could do gene therapy just through like an infusion, something a lot simpler. I think orally taking it by mouth as like a milkshake is probably not going to happen because of the digestive system. But are there other ways to do this? So it's like you go into the clinic, you get your infusion, you go home. Make something that's a lot simpler and faster and safer. So I think there's a lot of effort happening in that direction right now. It's another key milestone for us. There's a lot of looking at other ways of treating Parkinson's through the gut, though. We heard some of this at the symposium. What was it, last week ago? Okay. Any other questions I'm missing?
It's like one just came in.
So what was said is the only proven way to slow progression is exercise. So is there any way to understand what is occurring in the dopamine system when you exercise? And there actually has been a lot of research done on what's going on with exercise. The brain is not muscle tissue, but it operates a lot like muscle. The more you use it, the healthier it stays. That's been proven not just for Parkinson's, but for just about every ailment of neurodegeneration. So go ahead.
So I love this question because this is like near and dear to my heart. One of the things that happens with exercise is it causes growth factors to be released. And one of them is GDNF, which is one of the drugs that we're studying. And it's one way for the cells to say, we're healthy, we're okay, keep growing, stay alive. And so I think that is actually one reason why we've seen such benefit with exercise. It releases these really healthy, healthful growth factors into the parts of the brain that need it.
But important part here is, while it can slow progression, it's clearly not stopping the disease, and that's where the gene therapy comes in. It's like we've learned about these growth factors and everything from lab studies and studying people exercise, and looking at what's different in a Parkinson's brain versus a non-Parkinson's brain. The gene therapy is basically taking those effects and concentrating it. It's a boost. The exercise isn't necessarily going to get you enough GDNF to really put it over the edge, but the gene therapy might add to that. And then everything is just, as we said, the more you exercise, the more you think, the more you use your brain, you're reinforcing that gene therapy. And now we may actually see some true excessive benefit.
It's also possible that exercise alone may not be enough if Parkinson's has moved far enough along that you need more of a boost. Yeah.
Right. And that's, again, where the gene therapy comes in, is if you're already over that hump where exercise just isn't enough. That's where the medicine can come in.
Yep.
So this was brought up last week, and I did look into this a little bit. I have to say, this is a reach. There's nothing really solid here. So this is actually based on, again, older research. This whole idea that dopamine is causing the degeneration, dopamine is causing Parkinson's, and the thing that doesn't make much sense here is that tyrosine is simply a precursor to dopamine. We're talking about, instead of using L-DOPA that goes straight to dopamine, we're giving more tyrosine that goes into L-DOPA that goes into dopamine. So it's the same idea of you're just building up the well so that dopamine more readily made. It's not addressing the issue of dopamine being toxic. Dr. Bernstein's done a lot of great things. He is not a neurodegeneration doctor by trade. He is kind of a little bit out of his element when he's talking about this. So I don't want to completely discount it, but it's nothing new, and it doesn't actually answer the problem he claims it's answering. So
I just want to add, I mean, maybe we can do a talk once on dopamine may be part of the problem. I'm a big believer.
Yeah, we're not saying
dopamine contributing. Yeah. And so I think that may be an interesting topic to bring up at some point.
Yeah. So I think we should talk about that in detail. We can also bring up Dr. Bernstein's work. Well,
and just in general, if there's other topics that you guys would like us to dig into, we are really open, all ears. One thing I was thinking of trying next is talking about some unusual or weird symptoms that can be associated with Parkinson's. I think sometimes people forget or even afraid to mention some symptoms that they may be experiencing because it seems so weird. But I thought that might be a fun topic to try as well. But please reach out to Casey if there's anything that in particular that you would really like us to dig in on.
Yes, please do.
Yeah,
well, it looks like there's another few questions, and then we'll conclude.
There's one really, really interesting question that just came through. So let's start with the first one here. How soon do you see some form of gene therapy being implemented? I mean, there's some for other diseases, but what about Parkinson's disease?
It's going right now. So
there are trials going. So
yep.
We are still talking years down the road, but so we don't have a crystal ball to say exactly when, but things are in progress. Now, the next one I find really interesting. This is something I've actually wanted to study. Despite being an athlete in my youth, whenever I ran, I never felt a runner's high. Could that be a precursor symptom?
I have no idea. My initial gut reaction is no. but I don't think we really know. What were you going to say?
Well, no, it's just this idea of, and it's been looked at some other things. I was actually talking to you about this the other night, where there was some older research suggesting people that have a more hyperactive youth or a higher metabolism
Yeah.
are more prone to Parkinson's disease. So it's this idea of, could any mishandling of dopamine in youth, whether that be hyperactivity that burns the neurons out, I'm speculating here, or an already deficit of dopamine that prevents things like actually being excited on a roller coaster or after a good run. Could these be precursor symptoms? We don't know. A lot has been hypothesized. So obviously we can't say yes or no, but I think it's a really interesting thing that should be given more attention.
It's the right way of thinking. I think that's a really good kind of basis to come from. And again, this is what I was saying earlier, like we're starting behind the 8 ball. The horse is out of the barn by the time we're actually looking at patients. So, I mean, these are the questions we need to start asking ourselves. We need to get better at saying, can we follow someone out from their youth and say, this is someone who's destined to go on to get Parkinson's, and what are the differences that are happening there?
This is something I don't know about it. So, do you see it?
Yeah, Donna, is this what you had your hands raised for?
Yeah. Oh, I'm really loud.
I do not know this I guess chemical change. I'm not surprised. I think that this is why things like DBS exist, is that we need this more constant level of dopamine. There's a lot of reason to think that this is actually helpful in the long run, when things, instead of having big ups and downs, there's actually thought that contributes to the onset of dyskinesias. So I'll look into that. It's interesting. Thank you for sending.
I'm just concerned about the oral taking of levodopa long term.
Absolutely.
Really hurting you more than helping you. And they talked about in Dr. Bernstein's video about Cinemed holidays that I never heard of before. Old times they used to say, take time off from Cinemed and see if you improve.
I've had some patients do this. And it's different for everybody. And I think the reasons behind it are different, and what they experienced was different. But especially early on in disease, if you're just a couple years in, you may not notice much of a change for a while. But I'd say 100% people ended up going back onto Cinemed. But I think that it raises a very interesting idea. Maybe we should just have like a big dopamine Parkinson's discussion. Yep.
So just as a quick heads up, I just now looked at that paper real quick. So what they're saying is, for advanced PD, it was beneficial as far as signaling in the brain to switch from pills, so Cinemed taken out of schedule, to like the Duogel, so it's still levodopa, but the continuous pump.
There's actually,
The idea that they're going to develop a patch too, to be a more, we're forcing dopamine through the blood-brain barrier. So that's intracellular. Yet if the cells are dying off because there's too much dopamine inside the cell, and it's trying to regulate the thing that turns the dopamine production off so the cell doesn't die, and then I'm thinking, if you exercise, you're using that excess dopamine out of the cell. This is just conjectures I'm thinking about. But then
I love it.
So I think with all that in mind, just hold on to those thoughts. And I guess next month will need to be what's going on with dopamine.
But Claudia, I actually think this is a really good topic too. I had a couple people come up to me in the past, but not just at the symposium, about Lewy body, Parkinson's, that mix. That's another really good topic we should cover too.
Noted. Okay, well, thank you.
Thank you,
Dr. Van Laar's amazing information. My brain is overloaded, but in a good way.
Okay, good. It's a great way to start the weekend, right?
For
Yep.
sure. Hopeful.
Thanks, everyone.
Thanks. We'll see you next month, if not sooner. Be well.
Thank you.
Thank you. Thank you.
Thank you.
See
Bye-bye.
everyone. Thank you.
Bye.
Thank you. Bye.
Bye.